Hello, good afternoon students. Today we are going to discuss about the nitrogen fixation by blue-green algae which are non-symbiotic. Okay, non-symbiotic. Okay, so it is the non-symbiotic nitrogen fixation. Non-fixation. Okay, non-symbiotic nitrogen fixation. Last time we have discussed about the symbiotic blue-green algae nitrogen fixation. Okay, then now it is non-symbiotic nitrogen fixation by blue-green algae. So there are the different free-living nitrogen-fixing bacteria which are present in the air. Okay, so they grow in the marine water and the fresh water. Okay, so the free-living nitrogen-fixing bacteria. Here we are going to discuss Cyanobacteria, these blue-green algae, okay, which are marine as well as freshwater. Marine means they occur in the ocean and in the river or in the freshwater tank. So that is the freshwater, okay, or freshwater reservoir. म्हणजे नदी, तलाव वगैरे, ponds, beaches, freshwater, river, the freshwater algae. And the marine, it is in the ocean, समुद्रामध्ये त्या खाऱ्या पाण्यामध्ये असणाऱ्या ह्या ठिकाणी the cyanobacteria which are able to fix the atmospheric nitrogen, but they are not produce a symbiotic association, therefore they are non-symbiotic nitrogen fixer or free-living nitrogen fixing bacteria. Okay, so they are present in freshwater, marine and the freshwater sediment as well as in the soil, then leaf mold. ठीक आहे, म्हणजे पाल्यापाचोळ्याचा जो कचरा तयार होतो त्याच्यामध्ये किंवा इन द इंटेस्टिनल ट्रॅक्ट, in the intestinal tract of the some of the animals. Okay, intestinal tract of various animals. Okay, so काही फ्री लिव्हिंग त्या ठिकाणी नायट्रोजन फिक्सिंग बॅक्टेरिया आहे जे मराईन आहे, फ्रेश वॉटर सेडिमेंट मध्ये असणारे आहे. सेडिमेंट म्हणजे जो गाळा जमा होतो, फ्रेश वॉटर सेडिमेंट किंवा सॉइल मध्ये किंवा लीफ मोल्ड म्हणजे पालापाचोळ्याचा कचरा जो असतो लीफ मोल्ड किंवा इंटेस्टिनल ट्रॅक्ट ॲनिमलच्या इंटेस्टिनल ट्रॅक्ट मध्ये त्या ठिकाणी हे आपल्याला बॅक्टेरिया आढळतात. ओके, सो सम ऑफ द स्पेसीज दे आर एरोबिक, some of the species they are aerobic and some of the species they are anaerobic. Okay, so there are the two different types, aerobic and the anaerobic. Okay, so aerobic bacteria examples, that is azotobacter and the Bergeringa. Okay, so these are the examples of aerobic bacteria. Okay, and some of the species they are anaerobic and they can fix nitrogen only under very low oxygen pressure. Okay, म्हणजे काही जे आहे ते जेव्हा ऑक्सिजन लेवल खूप कमी असते त्या ह्याच्यामध्ये त्या ठिकाणी ते काय करतात, नायट्रोजन फिक्स करतात. ओके, मग या ठिकाणी याला म्हणायचं anaerobic bacteria which are non photosynthetic. Non-photosynthetic. Okay, the example is Clostridium, Bacillus and Klebsiella. And Klebsiella. Okay, so these are the different non-photosynthetic bacteria. And next, that is the aerobic or photosynthetic bacteria. What is the third one? Photosynthetic bacteria. Photosynthetic bacteria, and these are also known as cyanobacteria. Cyanobacteria. Okay, or these are also known as blue-green algae. Okay, so example is Anabina, nostoc, Lindia, Calothrix, etc. ओके, तर लक्षात घ्या आपण इथे नायट्रोजन फिक्सेशन बाय ब्ल्यू ग्रीन अल्गी पाहतोय, पण त्याच्या आधी थोडंसं आपल्याला माहिती असावं म्हणून तर या ठिकाणी नायट्रोजन फिक्सिंग बॅक्टेरिया. Okay, there are the several nitrogen fixing bacteria which may be non-symbiotic, okay, or which are free living, which are observed in marine water as well as in fresh water, in soil, in leaf mold and intestinal tract of the various animals. So this bacteria, it may be aerobic or anaerobic. So the aerobic bacteria which are non-photosynthetic, so these are the Azotobacter and the Weigerentia. Then anaerobic, these are also non-photosynthetic. Example Clostridium, Bacillus and Klebsiella. Then the third, that is, it is also the bacteria which is photosynthetic. Okay, photosynthetic bacteria. So this photosynthetic bacteria, they are known as the cyanobacteria. Okay, these are known as cyanobacteria. So both is aerobic, these examples non-photosynthetic, and the photosynthetic, they are known as the cyanobacteria. Example Anabina, nostoc, Lindia and Calothrix. These are the various blue-green algae. These are the various blue-green algae. Okay. So most of these free-living nitrogen fixers, okay, this shows the slow growth of the organism, and the non-photosynthetic species, they live in the habitat which is rich in inorganic carbon. Okay. Now we are going to discuss about the blue-green algae, cyanobacteria. Okay. So the cyanobacteria, it is the group of prokaryotic algae. Okay. The cyanobacteria, it is also known as blue-green algae. Okay. So these are the prokaryotic in nature. They are prokaryotic in nature and they found in fresh water. Okay, they are found in fresh water. It is found in fresh water. Okay, and the characteristic feature of this blue-green algae is this absence of membrane-bound cell organelles. Okay, absence of membrane-bound cell organelles. Okay. There is absence of membrane-bound cell organisms because it is the prokaryotic. Therefore, this membrane-bound cell organelles and the nuclei, prominent nuclei is absent because it is the blue-green. It is the prokaryotic in nature. Okay. So, and this blue-green algae, it has the presence of phycocyanin pigment. It has the phycocyanin pigment. So this phycocyanin pigment is the bluish green. Okay, it is bluish ग्रीन म्हणजे निळसर हिरवट रंगाचं असं त्या ठिकाणी पिगमेंट आहे सायकोसायनिन पिगमेंट. सो दॅट पिगमेंट इज प्रेझेंट इन ब्लू ग्रीन algae. Okay, they are occurred in unicellular form. For example, there may be they may be the unicellular form, or they may be the in filamentous form. Okay, or they may be in colonial form. Okay, the colonial form means they form the colonies. Filamentous, they form the number of cells they are lying one above another, they produce the filament, or they may be a single cellular. They may be unicellular or filamentous or colonial. Filamentous or colonial form. Okay, and they are enclosed in a mucilaginous sheath. म्हणजे याच्यामध्ये जर आपण या ठिकाणी पाहिलं, सपोज आपण नॉस्टॉकची जर या ठिकाणी कॉलनीज पाहिले, तर आपण या ठिकाणी हे नॉस्टॉकचे जर आपण या ठिकाणी कॉलनीज जर पाहिलं, तर या ठिकाणी हे कॉलनीज त्या ठिकाणी कसे असतात, दे आर बाउंड इन अ म्युसिलेजिनस शीट. ओके, so these colonies, they are found colonial form, and most of these, it may be the filamentous form. Okay, it may be filamentous or unicellular, or it may be the colonial. They have the mucilaginous layer, or these organisms, they are enclosed in the mucilage. Okay, they are enclosed in and most are enclosed in mucilaginous sheath. Okay, mucilaginous sheath. So the certain prokaryotes, they have ability to fix the atmospheric nitrogen by converting it into the ammonia. Okay, म्हणजे या ठिकाणी काही जे प्रोकॅरियोट्स जे असतात हे ब्ल्यू ग्रीन algae, तर या ठिकाणी त्यांची एबिलिटी काय असते, टू फिक्स द ॲटमॉस्फेरिक नायट्रोजन. म्हणजे ॲटमॉस्फेरिक नायट्रोजन, इट इज इन द मॉलिक्युलर फॉर्म. ओके, दॅट मीन्स इन N2 form, but it convert into the ammonia by the blue green algae. Okay, so some of the blue green algae, they are prominent nitrogen fixer in terrestrial as well as the aquatic system. म्हणजे हे काही जे ब्ल्यू ग्रीन algae असतात ते काही पाण्यामध्ये पण नायट्रोजन फिक्स करतात, म्हणजे aquatic system मध्ये, and some they may fix nitrogen in the on the land. Okay, so they are terrestrial. So some of the blue green algae, okay, they are prominent nitrogen fixer. Nitrogen fixer, they convert it into ammonia, NH3. Okay, and they fix terrestrial, as well as in aquatic system. Okay, they fix nitrogen. Okay, they fix nitrogen in terrestrial as well as in the aquatic system. Okay, so this nitrogen, they may be in the free living state, or it may be in the symbiotic role. Okay, they fix the nitrogen, it may be in the free living state or it may be in the symbiotic state. Okay, so most of the filamentous blue green algae, they have a specialized cell that is known as the heterocyst. Okay, मग या ठिकाणी हेटेरोसिस्ट काय असते, फॉर एक्झाम्पल नॉस्टॉक. ओके, so nostoc, the body is filamentous, but some of the cells which are empty and thick walled, which are larger in size, okay, which separates from the other cells. Okay, so it is the, suppose it is the non-stop. Okay, so these filamentous algae, they have a cell that is known as heterocyst. Okay, so this heterocyst, it differentiates the blue green algae from the other cells. Okay, this other cells पासून या ठिकाणी याचं फंक्शन वेगळं असतं. याचं फंक्शन काय, तर या ठिकाणी it carried out the function or nitrogen fixation under the aerobic condition. Okay, मग हे aerobic condition मध्ये nitrogen fixation कसं होतं, so this nitrogenase enzyme, it is present inside the heterocyst. Okay, this nitrogenase enzyme and the proteins required for nitrogen fixation, they are present in heterocyst. Okay, so there is, in the heterocyst there is a lack of photosystem second. Okay, photosystem second काय आहे की या ठिकाणी आपण पाहतो की एंजियोस्पर्मी प्लांट्स मध्ये फोटोसिस्टम फर्स्ट, फोटोसिस्टम सेकंड हे दोन सिस्टीम असतात. त्याच्यामध्ये फोटोसिस्टम सेकंड जे आहे, इट इज रिस्पॉन्सिबल टू रिलीज द ऑक्सिजन. ओके, आणि हे जे नायट्रोजिनस एन्झाइम आहे आपल्याला माहिती आहे की दिस नायट्रोजिनस एन्झाइम, इट इज सेन्सिटिव्ह टू द ऑक्सिजन. So this nitrogenous enzyme, when it exposed to the oxygen, it get destroyed. Okay, so to protect this nitrogenous enzyme in the heterocyst, only the photosystem first is present. Only photosystem first is present. Okay, and the second is absent, which is responsible for release of the oxygen. Okay, फोटोसिस्टम सेकंड, त्याच्यामध्ये ऑक्सिजन रिलीज होत असतो यांच्या सायकल झेड स्कीम फोटोसिंथेसिस मधले लाईफ CO2 फिक्सेशनचे जे लाईट फिक्सेशनची त्या ठिकाणी जे झेड स्कीम असते, तर त्याच्यामध्ये त्या ठिकाणी ऑक्सिजन रिलीज होत असतो. म्हणून मग त्या ठिकाणी हे फोटोसिस्टम सेकंड, इट इज अब्सेंट इन द हेटेरोसिस्ट. ओके, सो इट इज द mature heterocyst, it produces the nitrogenase enzyme and the proteins that involve the nitrogenase, the nitrogen fixation. It does not contain the functional photosystem second. It contains only photosystem first to protect the nitrogenase enzyme which is present in the heterocyst. Okay, so this heterocyst, it keeps the oxygen rich surrounding. आता हे जे सेल्स आहेत सगळ्यांमध्ये फोटोसिंथेसिस होते, इथे ऑक्सिजन रिच surrounding this, but it also the heterocyst, it keep away this nitrogenous enzyme from this oxygen rich surrounding. Okay, then heterocystous blue green algae. तर या ठिकाणी these algae, they fix the nitrogen aerobically and microaerobically. ओके, ज्या algae मध्ये heterocyst त्या ठिकाणी आहे, तर त्या ठिकाणी ते काय करतात, एरोबिकली आणि मायक्रोएरोबिकली या ठिकाणी ॲटमॉस्फेरिक नायट्रोजन फिक्स करतात. ओके, मग याचे जे स्पेसिस जर आपण बघितले की एक्झाम्पल जर बघितले आपण मगाशी अनाबिना, नॉस्टॉक, परत या ठिकाणी आपण तिसरं पण त्या ठिकाणी पाहिलं होतं लिंगमिया, कॅलोथ्रिक्स, त्याचबरोबर सायटोनेमा. दीज आर द व्हेरियस अल्गीज. ओके, परत एकदा लिहू आपण, नॉस्टॉक, अनाबिना, then Cytonema. So these are the different algae which are responsible to produce the heterocyst. These are the algae which have the heterocyst. This heterocyst which is responsible to protect the nitrogenase enzyme and it helps with the nitrogen fixation. Okay. So some of the algae, blue green algae, they are the non-heterocyst. Okay, some of the algae, they are non-heterocystous. Okay, so they do not have the heterocyst in them, so they fix the nitrogen only under the microaerobic conditions. Okay, so the example, blue green algae without heterocyst. ओके, म्हणजे या ठिकाणी अनाबिना, नॉस्टॉक, सायटोनेमा हे सगळे काय हेटेरोसिस्ट असलेले. पण या ठिकाणी काही जे algae आहेत तर त्या ठिकाणी they do not have the heterocyst. तर एक्झाम्पल जर आपण पाहिलं, that is the Plectonema, okay, Plectonema, Oscillatoria, then Trichodesmium, okay. So these are the, able to fix the nitrogen only under the microaerobic conditions, okay. So there are the 5 strains of Gleocapsa. Gleocapsa नावाची या ठिकाणी आलेली आहे. GLOEO capsa. Okay, Gleocapsa. That it fixes the nitrogen in aerobic condition. Okay, so it is aerobic. Okay, it fixes the nitrogen in aerobic condition. And there are the 5 strains. ओके, 5 त्या ठिकाणी स्ट्रेन्स आहेत ग्लिओकॅप्साचे जे एरोबिक आहेत. आणि देर आर 3 स्ट्रेन्स ऑफ सिनकॉकस. ओके, सो इट परफॉर्म द फिक्सेशन ऑफ नाइट्रोजन इन अनएरोबिक कंडिशन. ओके, अनएरोबिc condition. Then the mechanism of nitrogen fixation. Mechanism. Okay. So non-symbiotic nitrogen fixation takes place by the photosynthetic bacteria, that is the Cyanobacteria. So nitrogen is reduced by the electron of water. First of all, the non-symbiotic म्हणजे जिथे या ठिकाणी असोसिएशन नसतं, ते फ्री लिव्हिंग असतं. सो नॉन सिम्बायोटिक नायट्रोजन फिक्सेशन takes place by the number 1 Cyanobacteria, or which are also known as the photosynthetic bacteria. Like example, Nostoc or Anabina. Okay, so in this case the nitrogen is reduced by electron. Okay, so nitrogen is reduced by electrons of water. Okay, then second, that is the colorless saprophytic bacteria. Colorless saprophytic bacteria or green and purple sulfur bacteria or non-sulfur photosynthetic bacteria. Okay, the second, that is colorless. Colorless means non-photosynthetic. Okay, saprophytic, dead matter वर वाढणारे saprophytic bacteria or green and purple sulfur bacteria or non-sulfur photosynthetic bacteria. लाईक आता कुठल्या तर या ठिकाणी रोडोसुडोमोनास, Pseudomonas or Chromatium. Okay, or the Chlorobium, or Methanococcus. Okay. So where nitrogen is reduced by electrons. Okay. In this case, the nitrogen is reduced by electrons. It forms sulfur. Sulfide, hydrogen, and organic compound. Okay. So this second type, there is a colorless saprophytic bacteria, or the green and purple sulfur bacteria, or non-sulfur photosynthetic bacteria, like Rhodopseudomonas, Chromatium, Chlorobium, or Methanococcus. So they reduce the atmospheric nitrogen by the electrons from sulfur, sulfide, hydrogen, or organic compound. It converts into the ammonia. Okay. Then the third, the cyanobacteria, here photosynthetic है, हे colorless, non-photosynthetic है. Now colorless bacteria like Azotobacter and Clostridium. आता या ठिकाणी हे third जे आहे, तर या ठिकाणी these are the colorless bacteria. Colorless bacteria like Azotobacter or the Clostridium. They can fix the atmospheric nitrogen in dark with the help of ATP. Okay, they fix nitrogen in dark with the help of respiratory power, ज्याला आपण म्हणतो, with the help of ATP. Okay, ATP which is produced in the respiration. Okay, then the fourth one, that is free living saprophytic or photosynthetic bacteria. Okay, free living saprophytic or photosynthetic soil inhabiting bacteria, किंवा ज्याला सायनोबॅक्टेरिया किंवा अनएरोबिक वगैरे असं त्या ठिकाणी म्हणतात. ओके, सो free living saprophytic or photosynthetic free living saprophytic or photosynthetic soil inhabiting bacteria. Inhabiting म्हणजे soil मध्ये राहणारे. Photosynthetic bacteria which live in soil, or the cyanobacteria or either anaerobic faculty or aerobic bacteria. Okay, so the anaerobic conditions, they are the desirable because enzyme nitrogenase is highly susceptible to the oxygen. Okay, so here this nitrogenase enzyme, it is shielded in the protective covering. Okay, protective covering मध्ये ते nitrogenase cover केलं जातं. So it is with the help of nitrogenase is covered in the shield. Okay. Then aerobic and facultative bacteria, it fixes the nitrogen under anaerobic condition. Then aerobic nitrogen, it fixes cyanobacteria. They have thick walled heterocyst. Okay. Or aerobic cyanobacteria, they have the heterocyst inside which the nitrogenase enzyme is protective. Okay, and so they protect the nitrogenase enzyme in the helps to fix the atmospheric nitrogen. Okay, so if the heterocysts they are absent, the cyanobacteria they can fix the atmospheric nitrogen if the fields they are flooded with the water. Okay, and therefore oxygen is not available there. Okay, म्हणजे या ठिकाणी जर हेट्रोसिस्ट काही algae जर हेट्रोसिस्ट नसेल तर जेव्हा वॉटर फिल कंडिशन आहे त्या कंडिशनमध्ये तिथे ऑक्सिजनची लेवल आपोआप कमी होते आणि त्या कंडिशनमध्ये काही बॅक्टेरिया जे ॲटमॉस्फिअरिक नायट्रोजन फिक्स करू शकतात. ओके, देन the importance of blue green algae. Of blue green algae. Okay, so first importance, they act as a biofertilizer. They act as a biofertilizer due to their nitrogen fixing ability. Nitrogen fixing ability. Okay, and they successfully grow in the habitat with low or no combined nitrogen. म्हणजे ज्या ह्याच्यामध्ये ज्या सॉइलमध्ये अतिशय लो नायट्रोजन आहे किंवा त्या ठिकाणी अतिशय कमी प्रमाणात कंबाईन त्या ठिकाणी नायट्रोजन त्या ठिकाणी नाही अशा सॉइलमध्ये हे ॲटमॉस्फिअरिक नायट्रोजन फिक्स करतात. ओके, देन दे नॅचरली फाउंड इन पॅडी फील्ड. They naturally found in the paddy field. Okay, and improve soil fertility. Improve soil fertility and texture of soil. And texture of soil. Then these biofertilizers are environment friendly. Environment friendly म्हणजे जे आपण इको फ्रेंडली असं म्हणतो की it do not damage to the environment. Okay, so then they are the cheap alternative to inorganic fertilizers. म्हणजे इनऑर्गॅनिक फर्टिलायझर केमिकल फॉर्म मध्ये जे इनऑर्गॅनिक फर्टिलायझर त्याच्यापेक्षा हे स्वस्त असतात. therefore they are the cheap alternative because they have low cost in input. Because they have low cost input. Okay, input त्या ठिकाणी कमी असते. Okay, and they show effect after they show cumulative effect after 3 to 4 years. ओके, 3 ते 4 वर्षानंतर त्या ठिकाणी ते परिणाम दाखवायला लागतात. तोपर्यंत फार त्या ठिकाणी फरक जाणवत नाही. ओके, द मेन ॲडव्हान्टेज ऑफ BGA blue green algae is a biological nitrogen fixation. Okay, so it enriches soil with organic matter. It enriches soil with organic matter and reduces CN ratio. Okay, reduces CN ratio. Okay. Then other uses of blue green algae, they are enhanced the solubilization of immobile phosphate. Okay, they pass झाले. Okay, they are acted or they are used as a biofertilizer because they have nitrogen fixing ability. They are naturally found in paddy fields म्हणजे तांदळाच्या शेतात. They improve soil fertility, texture of the soil, environment friendly, cheap alternative to inorganic fertilizer because they have low cost input म्हणजे कमी खर्च लागतो show cumulative effect after 3 to 4 years, enrich soil with organic matter and reduces CN ratio. Okay. Then another uses, the sixth important, it enhances solubilization of immobile phosphate. Enhances solubilization of immobile phosphates. Immobile phosphates. Okay. And producing growth promoting substances. And producing growth promoting substance. The growth promoting substance वेगवेगळ्या प्रकारचे हार्मोन्स. ओके. ग्रोथ promoting substance in soil. Okay. Next importance काय? It enhances solubilization of immobile phosphate. म्हणजे फॉस्फेटची टेंडन्सी असेल जमिनीमध्ये फिक्स होण्याची, तर तो मग या ठिकाणी इमोबाईल होतो. मग त्याला सोल्युबलाइज केलं तर प्लांट त्या ठिकाणी ऍब्सॉर्ब करू शकतात. म्हणून इट एनहान्सेस द सोल्युबलायझेशन, ओके, ऑफ द इमोबाईल फॉस्फेट्स, and it produces a certain growth hormone or growth promoting substances in the soil. Okay, so which helps the plant to improve its growth. Okay, so they improve the physical, chemical and biological nature of soil. They improve physical, chemical and biological nature of the soil. And biological nature of the soil. Okay, and they are responsible for the long-term fertility. Responsible for long-term fertility. Okay, then it reduces the pH of soil and helps soil to exchangeable calcium. Okay, it reduces pH of soil. Okay, and it helps soil to retain exchangeable calcium. Okay, and help soil to retain exchangeable calcium. Okay, म्हणजे कॅल्शियम हे जमिनीतून प्लांट मध्ये ऍब्सॉर्ब होण्याची कॅपॅसिटी जी आहे ते वाढवतं, पीएच त्या ठिकाणी कमी करतं. सो दिस इट इज द इम्पॉर्टन्स ऑफ ब्ल्यू ग्रीन अल्गा. ओके, देन द प्रोडक्शन मेथड ऑफ बीजीए. Production method for BGA. Okay, so most commonly used species in biofertilizer production is the Anabina, Nostoc, Alocera and the Tulipothrace. Okay, so Anabina, Nostoc, Alocera and Tulipothrace. These are the various species which are of the blue green algae which are cultured. Okay, so pure culture of this blue green algae, they are transferred to the culture form. Okay, so the pure culture of these algae, okay, pure culture of these blue green algae, they are transferred to culture flask having the Fox medium, having फॉक्स मीडियम. ओके, जिथे फॉक्स मीडियम ज्याच्यामध्ये फॉक्स मीडियम आहे अशा कल्चर फ्लास्कमध्ये हे प्युअर कल्चर जे आहे ते काय केलं जातं, ट्रान्सफर केलं जातं. ओके, ट्रान्सफर. अँड it provided with the sufficient light. Provided with sufficient light. Provided with the sufficient light. Okay. Now this algal culture. आता या ठिकाणी हे जे algal culture या ठिकाणी ते तयार झालं. हे याच्यामधलं. It produced the algal culture. Okay. It produced the algal culture. So this algal culture, it is used for the mass production. It is used for the mass production. Okay. So there are the 4 different methods by means of which the mass production it can be taken. Okay. So these 4 methods, it may be the tank method, or it is also known as TROUG, T-H-R-O-U-G-S, TROUG method. Okay. Then second, it may be the peat method. The third, field method. And fourth, nursery cum algal production method. Nursery cum algal production method. Cum algal production method. Okay. So tank method, peat method, field method, and nursery cum algal production method. Okay. So one of the method we can use for production of the blue-green algae. Okay. So first of all, out of this, this blue-green algae, they are used for the algal culture. So pure culture is transferred to the culture flask having the fog medium and provide the sufficient light. The algal culture will produce, and this culture is used for the mass production. This mass production, it can be carried out by 4 methods. That is tank method or TROUG method or peat method, field method, or nursery cum algal production method. Okay. Now the mass culture, okay, or production method of blue-green algae. So first of all, prepared a cemented tank. Okay. Mass production. Mass production. So first, prepare a cemented tank. Cemented tank. Okay. Or shallow trays of iron sheet or polythene lined pit. Okay. Or shallow trays of iron sheets or polythene lined pits. ओके, यापैकी कुठलं पण एक या ठिकाणी आपण घेऊ शकतो. सिमेंटेड टँक घ्या किंवा शॅलो ट्रेस घ्या किंवा पॉलिथिन लाईन पिट्स, म्हणजे पॉलिथिन ज्या खड्ड्यांमध्ये असं पॉलिथिन अंथरलेलं असतं ते पॉलिथिन लाईन पिट्स. ओके. तर या ठिकाणी दिस पिट्स in an open area. Okay. But width of the tank, म्हणजे टँकची अशी जी जाडी आहे, सपोज हे टँक आहे, so it is the width. Okay. So this width, it should not be more than 1.5. Should not be more than 1.5 meter. Okay. Then transfer Then transfer 2 to 3 kg of soil. 2 to 3 kg of soil plus 100 gram superphosphate. Okay, superphosphate plus water. It is add in the tank up to 10 centimeter. Okay. Okay, 2 to 3 kg of soil plus 100 gram superphosphate plus water in the pit, and it is about 10 centimeter height. Okay, and mix lime to adjust pH. Mix lime to adjust pH. Okay, then add 2 ml of insecticide. Add 2 ml of insecticide. Insecticide. Okay. आता या ठिकाणी हे कशासाठी? To protect the culture from mosquito. Okay. To protect culture from mosquitoes. Okay. And it is mixed well and allowed to settle the particle. Mix well and allow to settle. Mix well and allow to settle the soil. Okay, soil त्या ठिकाणी settle होईल. Okay, when the temperature when the water it become clear, sprinkle 100 gram of starter culture. Sprinkle 100 gram starter culture. Okay, starter culture on surface water. On surface of water. Okay, so when temperature it remains around 35 to 40 degree centigrade during the summer. Okay, so in that case the maximum growth of cyanobacteria takes place. म्हणजे 35 ते 40 डिग्री टेम्परेचर जेव्हा असतं समरच्या टायमाला किंवा उन्हाळ्यामध्ये, त्यावेळी सायनोबॅक्टेरियाची मॅक्झिमम ग्रोथ त्या ठिकाणी होत असते. ओके, सो this allowed to grow. Okay, allow to grow. Okay, then maintain 1 centimeter water. ओके, म्हणजे कमीत कमी 1 सेंटीमीटर वॉटर त्या ठिकाणी राहिलंच पाहिजे, हे आपल्याला मेंटेन करायचं. ओके, देन आफ्टर ड्राइंग द algal mass it separate. After drying separate फ्लेक्स म्हणजे काय? ते हिरवे हिरवे जे फ्लेक्स ऑफ algal mass. Flakes of algal mass from the soil. Okay, and then it is collected, then well dried and powdered. Okay, and packed in polythene bag. Okay, and it is supplied to the farmer. Okay, it's supplied to the farmer. So this algal flakes, it can be stored in the or it is used as a starter inoculum again. Okay, so it should be stored in the cool and dry place. Okay, so we can store this algal powder for 3 years without losing its efficiency. Okay, so for 1 hectare of flooded rice or paddy field, we can use 10 kg of this blue green algae powder. Okay, 10 kg per hectare. Okay, so application of blue green algae to the crop, it is known as the algalization, and it yields, it increases the yield up to 34%. Okay, म्हणजे या ठिकाणी 34% yield जे येते, yield increases by applying 10 kg per hectare paddy field. Okay, paddy field. So it is the mass production of the algae. हा एकदा परत रिवाइज करू. पहिल्यांदा या ठिकाणी सिमेंटेड टँक किंवा शॅलो ट्रे किंवा पॉलिथिन बॅग ची कव्हर केलेले खड्डे तयार करायचे. तर त्या ठिकाणी याची विड्थ या ठिकाणी 1.5 मीटर पेक्षा जास्त असू नये. तर असे पिट्स ओपन एरियामध्ये आपण तयार करायचे. ओके, ओपन एरियामध्ये तयार करायचे. त्याच्यानंतर त्याच्यामध्ये 2 ते 3 किलो सॉइल टाकायची, 100 ग्रॅम सुपर फॉस्फेट टाकायचं, वॉटर टाकायचं जे जवळपास 10 सेंटीमीटर असेल. त्यानंतर पीएच ॲडजस्ट करण्यासाठी लाईम टाकायचं. त्यानंतर 2 ml इन्सेक्टिसाइड टाकायचं मॉस्किटोपासून प्रोटेक्ट करायला. and then mix well and allow to settle the soil. ओके, सॉइलला सेटल होऊ द्यायचं. सॉइल ते वॉटर क्लिअर झाल्यानंतर त्याच्यावर 100 ग्रॅम स्टार्टर कल्चर जे आपण फॉक्स मेथडवर तयार केलं होतं, कल्चरवर तयार केलं होतं, तर त्या ठिकाणी ते कल्चर त्या ठिकाणी टाकून द्यायचं 100 ग्रॅम. ओके, आणि त्यानंतर त्या ठिकाणी त्याला ग्रो होऊ द्यायचं आणि ॲटलिस्ट 1 सेंटीमीटर वॉटर त्या ठिकाणी मेंटेन करायचं. त्याच्यानंतर त्या ठिकाणी जेव्हा ते सॉइल वाळेल आणि अल्गल फ्लेक्स त्याच्यावर तयार होतील ते कलेक्ट करायचे, वेल ड्राय करायचे, पावडर करायचे आणि पॉलिथिन बॅगमध्ये पॅक करायचे आणि देन सप्लाय टू द फार्मर्स. ते फार्मर्स त्या ठिकाणी त्यांनी जर 10 kg पर हेक्टर जर पॅडी फील्डला या ठिकाणी हे अप्लाय केलं तर त्यांचं यील्ड जे आहे राईसचं 34% इन्क्रीज झालेलं दिसून येईल. ओके, सो आय होप यू गॉट द पॉईंट्स. थँक्यू व्हेरी मच.